and water will rush into the cell and cause it to swell up. And that's the first main change of cell injury. It's still reversible. We'll talk about the reversible changes first, but it's not very good. So the cell will swell up because the sodium can't leave, and even some of the organelles within the cell will start to swell. Like the rough ER will swell, ribosomes will detach. Then what do ribosomes do? They make protein, right? So you will lose the ability to make proteins. That's no good. And what type of metabolism do you think the cell will start to undergo? Actually, we'll start to perform glycolysis. That's not very good either. Because without any oxygen around, that's the only way it can make ATP. How many ATP per glucose molecule do you think it'll make? About 2. Whereas with aerobic respiration, it was making 38. So 2 is only a fraction of the amount that it could make with oxygen. That's terrible. But it's all it can do, so it's just working with what it's got. All right. Another change we see is that the cell starts to turn pink or becomes hyalinized. And why is this? It's because of a drop in pH or acidification of the cell. And why does this happen? Well, guess what? To explain this, we have to talk about biochemistry again. What? You don't really mind? You sure? You actually kind of like biochemistry now? Wow. Okay. Well, we'll talk about it then some more. So, again, a drop in pH will actually cause the cell to turn pink, but also the nucleus to clump up like that. But here's the biochemistry snapshot. So, remember what I said, that there's no more flow of electrons anymore because there's no electron acceptor, so there's a stasis of electrons in these complexes. So that means that NADH won't be able to dump off any more electrons, and there will be a really large buildup in NADH and a decrease in NAD+. Now the problem with this is that NAD+ is actually required for glycolysis, some of those dehydrogenase reactions, and glycolysis, we need that to make our measly 2 ATP. Shoot. So we need to regenerate NAD+. Even though it's not very helpful, it's all we've got to make ATP. How are we going to do that? How is the cell going to do that? So to answer that, what is the final product of glycolysis? Pyruvate. Okay. And guess what? When pyruvate actually gets converted into something, NAD+ is regenerated. What does it get converted into? Lactic acid or lactate. The problem with lactic acid is that it's an acid. Oh no. So that's why we get the drop in pH. So now it all makes sense. So we get NAD+ and we keep making ATP, but at the cost of acidifying the cell. All right, there's another reaction that actually regenerates NAD+. And do you know what that is? It's actually turning dihydroxyacetone phosphate, well, that's a mouthful, into glycerol 3-phosphate. So glycerol is actually the backbone of triacylglycerides. Believe it or not, when you string 3 fatty acids onto glycerol, you have yourself a triacylglyceride. And that's why there's a buildup in fat within the cell. So these are all of the reversible changes so far. Now